Synapse pruning is a biological necessity. Throughout early development and into adulthood, the brain actively eliminates weaker connections to optimize neural circuits. But in neurodegenerative disorders like Alzheimer's disease, this exact mechanism overcorrects, accelerating severe cognitive decline. Historically, neurobiology divided the study of synapse elimination into two separate pathways. One theory focused on the complement system, an immune response governed by glial cells. The other focused on neuronal pruning receptors, specifically PirB in mice, and its human equivalent, LilrB2. For years, researchers knew that a specific immune system protein called complement component 4, or C4, was a significant genetic risk factor for both schizophrenia and Alzheimer's. The prevailing model assumed C4 caused synaptic loss entirely by signaling microglial cells to physically engulf and digest the synapses. To understand the actual mechanism, we have to track the biochemistry. When the complement cascade is activated, a protein called C1q cleaves the C4 protein, breaking it down into a highly active fragment known as C4b. C4b is then further processed, shedding parts of its structure until it becomes C4d, a terminal split product that covalently bonds to the local cell membrane and remains fixed at the site of the initial immune activation. For decades, biologists classified C4d as an inert biological footprint. It was utilized in clinical settings as a diagnostic marker for organ transplant rejection, but it was thought to have no intrinsic signaling function of its own. A 2025 study from Stanford University identified C4d as the specific molecular bridge that connects the complement cascade directly to neuronal pruning receptors. The researchers discovered that C4d binds specifically to those exact neuronal receptors previously thought to operate in isolation, LilrB2 in humans and PirB in mice. The human receptor, LilrBr2, is a well-documented risk locus identified in multiple genome-wide association studies for Alzheimer's disease. This binding interaction directly connects genetic susceptibility to physical synapse loss. By identifying C4d as a functional ligand, this research unifies the glial engulfment theory and the neuronal receptor theory into a single, highly coordinated system where immune proteins directly issue chemical commands to neurons. To establish that this relationship drives synaptic pruning, the researchers first had to prove the physical chemistry. Does C4d bind to the LilrBr2 receptor with the specific kinetic strength required to trigger a cellular response? They tested this using surface plasmon resonance, or SPR. SPR is an optical technique that measures the real-time binding kinetics and affinity between two molecules by detecting changes in light refraction as they interact. The SPR analysis revealed a clear concentration-dependent response between soluble LilrB2 and purified C4d. The dissociation constant, or KD, was approximately 3 nanomolar, indicating an exceptionally high-affinity physical connection. To prove this interaction wasn't a generic immune response, they tested C4d against closely related receptor family members, like LilrB1. It showed almost zero binding affinity, confirming the interaction is strictly tailored to LilrB2 and PirB. Demonstrating affinity in a purified solution is only the first step. The molecules must interact reliably when the receptor is embedded in the complex environment of a living cell membrane. This chart measures the mean fluorescence intensity of C4d applied to cells expressing the mouse PirB receptor. The top curve shows a steep, saturable binding profile with a KD of 56 nanomolar, while the empty vector control remains flat at the bottom. This graph plots the binding isotherms for actual wild-type cortical neurons compared to those engineered without the PirB receptor. The wild-type neurons, shown by the top line, readily bind C4d. In the PirB-knockout neurons below, that binding capacity is significantly reduced. Receptors like LilrB2 and PirB are large proteins built with multiple extracellular immunoglobulin domains. The researchers needed to isolate exactly which segments of the receptor latch on to C4d. These Western blots show the pull-down results for several truncated receptor versions. Thick, dark bands indicate a successful capture. C4d consistently binds to constructs containing both D1 and D2 domains, identifying them as the minimum requirement for connection. The biochemical evidence establishes C4d as a potent, high-affinity ligand. It physically targets specific structural domains on the exact neuronal receptors known to drive synaptic proning. If this mechanism drives disease, the proteins must physically occupy the correct anatomical locations. The researchers examined human prefrontal and temporal cortex tissue to see if C4d and LilrB2 are stationed at synapses where pruning occurs. Mapping proteins at a synapse requires overcoming a major technical barrier. Standard light microscopy cannot resolve individual synaptic compartments without significant optical blur from proteins positioned immediately above or below the focal plane. To solve this, the team used array tomography, an imaging technique where brain tissue is embedded in resin and sliced into 70 nanometer sections, allowing for spatial identification of proteins without vertical background noise. Panel A displays excitatory synapse markers alongside LilrB2, C4, and C4d, showing immune proteins overlapping in the neuropil. In Panel B's synaptogram, tracking a single synapse through 4 slices reveals LilrB2 and C4d perfectly overlapping in Section 3. Panel C's Pearson correlation analysis proves the overlap between LilrB2, C4d, and the synapse is physical reality, not random chance. Panel B quantifies this. Nearly 60% of excitatory synapses showed C4 and LilrB2, and 30% contained C4d. These Western blots provide biochemical confirmation. By separating brain tissue to isolate synaptosomes, the isolated synaptic terminals, researchers found heavy concentrations of both LilrB2 and C4d right where the synapses connect. When analyzing samples from patients with advanced Alzheimer's disease, the data showed a distinct localized decrease in both 95 levels per synapse, tracking the physical loss of connections as the disease progresses. In the Alzheimer's samples, LilrB2 and C4d heavily co-localized with beta-amyloid oligomers, particularly at excitatory synapses located immediately adjacent to dense amyloid plaques. While protein counts naturally shift during aging, total C4d protein levels were elevated 4-fold in the prefrontal cortex of Alzheimer's patients compared to healthy controls. The anatomical data places C4d and LilrB2 at the exact scene of the crime. They are physically anchored to the excitatory synapses of the human brain and highly concentrated precisely where Alzheimer's pathology does its worst work. Binding affinity and physical location prove the mechanism is possible. But the ultimate requirement for biological causation requires answering one direct question. Does elevated C4d actually drive the elimination of dendritic spines? To test this in vivo, the researchers designed a gain-of-function experiment. They surgically implanted osmotic mini-pumps into mice to continuously infuse pure recombinant C4d directly into the primary visual cortex over 4 days. For visibility, they utilized Thi1-YFP-H mice. These mice are genetically engineered so specific neurons express a yellow fluorescent protein, giving researchers a map to count dendritic spines before and after infusion. In wild-type mice, C4d exposure triggered a rapid decrease in dendritic spine density. However, when repeated in PirB-knockout mice, lacking the receptor, spine density remained indistinguishable from controls despite the C4d flood. The severity of the spine loss in the wild-type mice matched the diffusion radius of the mini-pump. Neurons within 800 microns of the infusion site lost spines, while neurons safely outside the chemical radius maintained normal densities. This total rescue in the knockout mice proves specificity. If C4d was simply triggering a general inflammatory microglial attack, the lack of a neuronal receptor would not have prevented the loss. The synapse loss required the PirB receptor to execute the command. This aligns with previous functional observations. PirB-knockout mice bred to model Alzheimer's disease display high beta-amyloid loads, but they do not experience the cognitive impairment or memory loss seen in normal mice with the same plaques. Furthermore, because C4d is a terminal cleavage product, it lacks the domains to reactivate the downstream C3 glial engulfment pathway. The spine loss we see here is isolated to the C4d-PirB interaction. Natural C4d levels correlate with age, increasing in both human and mouse cortices over time. This suggests the C4d receptor axis contributes to the normal age-related cognitive decline experienced by the general population. From a pharmacological standpoint, the LilrB2 receptor presents a druggable target. Blocking this specific interaction could prevent pathogenic synaptic loss without requiring doctors to fully suppress the immune system's glial functions. The in vivo data provides the final proof. Elevated C4d acts as a direct chemical kill command for synapses, but is powerless unless the neuronal LilrB2 or PirB receptor is there to receive it. The data yields a dual effector model of synaptic pruning. When the complement cascade activates, it splits its attacks. C4b engages the glial cells to consume synapses, while the terminal C4d fragment binds to neuronal receptors to trigger structural retraction. This discovery highlights a major limitation in current Alzheimer's research. Drugs designed exclusively to suppress glial activation and target immune receptors are only blocking half of the pruning process. Identifying a specific neuronal pruning receptor provides a new vector for therapy. By targeting LilrB2 directly, pharmacologists have a precise mechanism to preserve neural architecture and protect motor and memory performance in the aging brain. This finding from Stanford University forces a reevaluation of how the nervous and immune systems communicate at the molecular level. While this study prioritized Alzheimer's and aging, the established link between C4 and schizophrenia suggests this C4d receptor interaction may provide a mechanism for investigating the synaptic changes seen in other neurodevelopmental disorders. Confirming C4d as a functional ligand establishes the immune system as a direct regulator of synaptic density, providing a precise molecular map for therapeutic intervention.